Afternoon, everyone. Thank you for joining us today for this monthly MMI Advanced Microscopy seminar. I am coming to you today from the lands of the Wurundjeri and Burunong people of the Kulin Nation, and I invite everyone to take a moment to pay respect to the traditional custodians of the lands and waters on which they currently stand. It is my pleasure today to introduce Professor Jan Huisken. Jan is a Alexander von Humboldt professor at the Georg-August University in Göttingen. He's best known for his work in his interdisciplinary work at the interface of gentle high-resolution microscopy and quantitative developmental biology. After initially studying physics, he pioneered multidimensional light sheet microscopy that you might know as SPIM, so selective plane illumination microscopy, in Heidelberg. He then moved to Didier Stenier's lab at UCSF to apply this new technique to study the morphogenesis and function of the zebrafish cardiovascular system. He then became an independent group leader in 2010 at the Max Planck Institute for Molecular Cell Biology and Genetics. And his remarkable contribution to the field of microscopy has been recognized multiple times, particularly with the award of the Royal Microscopy Society Medal for Light Microscopy in 2017 and the Lennart Nielsen Award in 2020. His lab is now based in Göttingen since 2021, and they use advanced customized light sheet microscopy, so LSFM and SPIM, to image biological and samples at various scale, from small living organism to large fixed and clear tissue. So as we know, the experimental setup and optimization is critical to streamline this process. His lab has developed a modular and portable light sheet microscope framework named Flamingo. And Jan will today talk about this new system in his presentation titled Flamingo, Development, Application and Dissemination of Light Sheet Microscopy. Please join me in welcoming Jan to the Monash Advanced Seminar Series. And Jan, the floor is yours.
Yeah, thank you so much. Thanks for the invitation. Thanks for joining us today. So, as was already said, Flamingo is the main topic for today. So this is really an exciting project I want to report about. This is our latest light sheet microscope, and it offers a ton of opportunities for us nowadays. It's not just a small little portable microscope that we like to share with other people, but it's really a high-end instrument that we use every day in my lab. It's our workhorse, but it happens to be portable, and that opens up a lot of possibilities that we had never imagined, really. So in today's talk, I'll give you an introduction, sort of quickly who we are and sort of which direction we're coming from. Briefly introduce light sheet microscopy, which I assume most people are already familiar with. Give you a brief summary of some of the things we've done in the past, mostly in zebrafish, and then introduce the Flamingo concept to you, why we came up with this idea and how valuable it is for us in our everyday work, and why we think this could be an interesting technology for you. And if I have time, I'll briefly go also into clear tissue imaging and talk about the advantages of remotely controlling such an instrument. So just to introduce my lab a little bit. So we are a multidisciplinary lab, and I believe that's really the key to modern light microscopy development. We try to cover all these areas depicted here, which isn't easy, and we don't necessarily cover all bases, but obviously we also heavily collaborating with all sorts of other scientists that help us out with like data processing, for example, and we have great help from our animal caretakers for, let's say, the zebrafish husbandry. But in general, I like to gather all these people around one table in lab meeting to basically have this cross-fertilization where the biologists can dream big, come up with a dream experiment, and hopefully the physicist can design an instrument and build a microscope to enable the biologists to do things that they couldn't otherwise do, let's say, with commercial instruments. So if you, generally speaking, want to understand function morphogenesis in biological systems with quantitative imaging approaches, you basically end up trying to image things alive. You're trying to image whole organisms, embryos alive, and you have to do so very gently. So you're suddenly facing a lot of challenges in terms of sample mounting, image analysis, etc. And at least for the last, decade or two, there was absolutely no such instrument available. And so that really forced us to spend most of our time building our own microscopes. Nowadays, you can buy a commercial light sheet system, but these still are sort of general use instruments, and they may not be specifically designed for the kind of experiment that you're trying to do. And so what we end up doing is really trying to build the instrument around the sample. So basically not building a microscope like maybe depicted here on the left and then say like, oh, you have to mount your sample on a cover slip, otherwise it won't fit into the microscope, but basically have the sample dictate how close the microscope can be to the sample, and rather develop mounting techniques that are ideally suited for the sample, but may not be ideal for the imaging, but then still find ways around that. And so if you want to summarize what we do, we're basically trying to build the best possible microscope for a given application, or building the microscope around the sample. And so while these might be, some cartoons depicting this idea, this is really what drives us. And this is also where this multidisciplinarity is key, so that the microscopy developers are not just sitting in a dark room, building fancy microscopes that maybe nobody needs, but actually develop things that push the biology forward and enable new experiments. So the issue, however, is that you end up with many custom-built microscopes. If you're trying to build the best possible instrument for each experiment, you end up having many microscopes. And of course, not just the people in the lab, but people outside the lab on campus or just anywhere across the world might have an interest in using your instrument. So if you want to be maximally collaborative, then you also have to think about how to share the technology with other people. Publishing a cool new microscope in whatever Nature Methods doesn't really help anybody if nobody is able to reproduce that system. And if you're basically asking people to come to your lab to use the instrument. So that's something we ultimately need to address if we want to have a maximal impact. So briefly, light sheet microscopy, from my perspective, is an incredibly valuable technique, which also is relatively straightforward to implement. So I think this cartoon summarizes quite nicely the core features of a light sheet microscope. So typically you have two perpendicular optical arms. One is used for illumination. You're projecting a thin light sheet, laser light sheet, into the specimen, and then perpendicular to this, you're collecting the fluorescence from the sample. And because you're illuminating only one plane, one thin section in the sample, you directly get optical sectioning, in a very gentle, in the most efficient way. You're only exposing a single plane in the sample, and you're not harming anything outside this plane of interest. At the same time, you can use a super fast, super sensitive camera in your detection path, and that really allows you to take high-speed videos of whatever you're looking at in this one particular plane. And of course, it also allows you to take volumetric data by simply moving the specimen through the light sheet as you're recording a video. And so within a few seconds, you can take a Z-stack, which on a confocal would take many minutes, if not, an hour or so. So essentially, the core values ultimately are then low phototoxicity and high speed. And there's a number of different flavors of light sheet. Nowadays, I'm not going to review all of these. We used to call it SPIM, selective plane illumination microscopy. Now it's mostly referred to light sheet fluorescence microscopy, or LSFM. Call it whatever you want. Ultimately, it comes down to these principles and these main advantages. And the beauty of it is also that it's very much scalable. So you can pick whatever lens you want, depending on the working distance and the magnification needed. You can scale this to the specimen. Ultimately, the specimen has to fit between the lenses, and the light sheet should have properties that allow you to get good sectioning across the entire field of view. So in this case, depicted here on the left, we have actually 2 illumination lenses, which helps getting better depth penetration on the illumination side, because you can illuminate from one side and from the other side, and thereby get a more complete image of your sample. So since the early days of light sheet microscopy, so my PhD work back at EMBL, together with Ernst Stalzer and Jim Swoger, since the early days, really not much has changed in terms of the core values. And if I want to list them, it would probably somehow be in this order, which again highlights that the phototoxicity ultimately is the main value for us. So there's no value in a microscope if it kills your specimen, right? You're imaging artifacts. You're not imaging your biological specimen under physiological conditions, and you're not recording true biology. So the most important is that the sample survives the imaging, simply speaking, and that's something light sheet is particularly good at. And so this is really our most important, and this is achieved by being maximally efficient in the excitation and the detection, and being fast and gentle. So of course, also light sheet gives you very good depth penetration and very good resolution, but these are not, the main design criteria. So we are not pushing for the ultimate resolution if that would sacrifice, the gentleness of the approach and would create artifacts in the imaging. The other thing that's listed here on the bottom, instant on, what I mean by this is that we really value instruments that are easy to use. And there's more value in having many simple microscopes than one super duper instrument that you need to align every single morning for an hour before a biologist can use it. So it's incredibly important that everybody in the lab can basically use any of these instruments very quickly. It's easy to learn to use these instruments, and it does not require major alignments every single morning. Otherwise, you're just not using the instruments as much as you should. So this has really been driving us in the past, and together with the biologists and our main interest in zebrafish morphogenesis, We've recorded lots of data, and I'm not going to go through all of this. It's all in the publications, but just to highlight a few things that we are most well known for. One of this is the zebrafish heart, imaging a beating, quickly moving organ in a whole living organisms has been a challenge that nobody had really achieved back in the days. And all of these data that I'm showing you here on the slide would have actually been impossible with any other microscope technique. So really, light sheet has enabled us to do a lot of things that were simply impossible. So here we see basically a 5-dimensional reconstruction, if you will, of a zebrafish heart. So this was recorded by imaging the heart in different planes at high speed in 2 colors, and then reconstructed by synchronizing these movies and reconstructing the heart in 3D. The next one, showcasing the low phototoxicity, is a 3-day time lapse of a developing zebrafish larvae, where we've highlighted not just the vasculature, but also the blood flow. And the beauty of this data is really that we can image the fish for so long. And at the same time, as you can see, we can even record it from different directions. And so I don't really have time to go into this, but you can imagine that if you're imaging something that's about 1 millimeter across and several millimeters long, you cannot image it with even high resolution from just one side. So it helps a lot if you can rotate the specimen, image it from different sides, and then reconstruct it with all of these data. And in order for this to work, you really need to be very fast. So that all of these different views will eventually line up nicely. So these are examples for high speed and low phototoxicity imaging. This gastrulating zebrafish embryo here on the top is also a reconstruction from 8 different angles. So I should say the fish embryo is about 1 millimeter across. So in order to image something at this level of resolution, you need to combine data sets from different angles. And what's really ultimately the challenge in all of these data is that you have sometimes terabytes of data, such a time lapse. This is like 24 hours time lapse. This can accumulate tens of terabytes depending on your resolution. And the challenge, of course, is then to process all this data and make sense of it, and not just image a handful of embryos, but ideally, a sufficient amount of samples that would allow you to draw some conclusions on how exactly does a fish develop and how much variance is there between one embryo and another. And then another example here in the top right, where we see these axons growing out and the lateral line forming. Another beautiful example showcasing the quality of the data. And I must again emphasize that the sample preparation is absolutely key to success here. And people spend a lot of time making sure that the embryo is still for the time period of imaging, but at the same time free to grow in order to let it expand and not be crippled by being embedded too tightly in a gel or something like this. And then on the bottom right, this is just a placeholder to also indicate that light sheet microscopy is also ideal if you are imaging cleared specimens. At some point during development, of course, the sample becomes too big, not translucent anymore, and then imaging the sample alive may be impossible. And at that point, you may need to fix and clear the specimen, but still another light sheet microscope with maybe a different set of lenses is still your friend and the best technique to do this kind of imaging. And imaging a large volume quickly is still something that only light sheet can do. So in another project, we've looked very carefully at the details of the zebrafish heart. So here we see on the right the raw data, a cross-section through the heart. On the left, a 3D reconstruction. And I don't really want to go into the details, but we were mostly interested in the mechanics of the heart. So how is the force that the myocardium exerts transmitted onto the endocardium? How is the endocardium folded to make this 3-layer pump an efficient pump? And we were able to reconstruct the heart very nicely and visualize not just the myocardium and endocardium, but also the cardiac jelly, and even reveal the details of how the endocardium is attached to the myocardium and allow it to fold in a very particular manner every single time the heart contracts, like 3 times a second or so. So this is the 3D reconstruction, and you can appreciate that from data like this, you can then reconstruct and draw a model of how exactly the mechanism of how cardiac contraction works in the zebrafish. And we've taken this even as an inspiration to understand how maybe efficient pumps can be engineered with which you want to pump a medium that isn't just a fluid, but also, like the blood cells, contains some particles that you don't want to destroy, you don't want to compress, where maybe a peristaltic pump would be too strong and may destroy the content of your liquid. And so there's some bio-inspired engineering that you can do based on these mechanisms that we can see in the heart. So I briefly mentioned, and again, don't have time to go into this, but just want to emphasize how important it is to actually visualize the data and represent the tracks that all these cells are taking. I showed you the gastrulating zebrafish, to visualize the data and to condense it in a more digestible form, so that maybe sometimes these 5-dimensional data sets need to be reduced to a 2-dimensional image. And this is shown here by condensing the tracks into the highways that the cells are taking, and then on top of that doing a Mercator projection, which basically visualizes the entire embryo in a world map, so to speak. And that can sometimes be the key to actually understand the data and be able to inspect the data very quickly after a long time lapse, where all these terabytes of data may just be very, very challenging to even look at and inspect to see if the experiment has been successful or not. So, and this is really also important for future developments, where we think a lot about these smart microscopes, where you essentially have to inspect the data in real time automatically to make choices whether you want to continue the experiment, you may want to change your area of interest, your region of interest, you may want to change the magnification, being able to zoom into particular portions of the image to be able to record more information and less overhead, and reducing effectively the amount of data that you're collecting. Just want to briefly mention that on top of the imaging, there's also a lot of sample manipulation that you may want to do. So for example, here we've injected magnetic beads into the embryo, and then with electromagnets, you can rotate the specimen in the sample preparation, and that allows you to reorient the embryo during a time lapse, and basically gives you more degrees of freedom to reorient the specimen and get the best possible angle on whatever you want to image. We've also done quite a bit of optogenetics, where you are manipulating the cardiac rhythm. So here, for example, we express halorhodopsin in the myocardium, and by illuminating the pacemaker, you can actually stop the heart and thereby then visualize or study processes in the zebrafish that depend on blood flow. And this is perfectly reversible, so you can turn off the light and the heart continues to beat. So all of these experiments were done on different microscopes. So every video that I've shown you was basically done on a different light sheet microscope. And as I said, you essentially run into this problem that you have many microscopes, and all of these instruments are built by different people in the lab. And as we all know, there's always some turnover. People come and leave, and so you end up with this problem that you have big optical tables full of optics built by some person who may have already left the lab, programmed in LabVIEW or whatever, and nobody in the lab really knows how this instrument works, and if it breaks, nobody would be able to repair it. And so I found myself in this situation when I moved from Dresden to Madison, Wisconsin, that we had all these wonderful instruments in the lab, but there was really no chance for us to ever move them to the new location. And that, of course, is a pity, because you've built all these fantastic instruments, you've spent a lot of money, and ultimately you're not able to use them anymore. And so that was the moment where I thought, well, this has to change. And again, there's a long list of things that a lot of us are probably facing, and most people here are probably familiar with the situation. And this would also hold true probably for some commercial instruments that you bought a few years ago, and maybe the person who knows how to run the system has left the lab, and now this expensive instrument sits there and collects dust. So microscopy developers have a lot of these home-built microscopes that are simply not sustainable. So it can be very wasteful to always just build a new instrument, use it for a few experiments for your publication, and then there's just no way for you to reuse it in some meaningful way just in your own lab. And so we thought it would be wonderful if we had a reproducible system, a microscope that we would feel comfortable reproducing, because we feel like this has been very nicely engineered. It's a system that we can use for many experiments, and that would have like a solid electronics and solid software platform that we could use for many such instruments. And of course, there's a great value of having many microscopes in the lab. It would basically allow you to have an instrument for every student or every postdoc in the lab, and so it increases your throughput and output. And as I said earlier, I see more value in having many microscopes than having just a single microscope that may be 5%, 10% better, but you only have one of these. So having many has a big value. And of course, it can increase your throughput. And if you are thinking about a lot of problems in developmental biology where you need high N, where you need to image a lot of samples, you also need many microscopes. At the same time, if the system can be adapted and changed, let's say if we could reproduce all the instruments that I've now shown you, using the same hardware and the same software would of course be phenomenal, because it would allow us to very quickly also adapt the system to different needs. And suddenly we would be able to image a much greater variety of samples, and we wouldn't be restricted to, let's say, the zebrafish or whatever we've optimized the system for. And so that's exactly what the Flamingo ended up being. And so I had have a wonderful team of people, and especially want to highlight these three guys who've developed the optics, hardware, electronics, and software. And It is really an instrument that we've never built before. Something where you really also, as a scientist, it's this challenge of making this call where at some point you say like, yep, that's it. This is a great instrument. We now want to build many of these. It's not the usual thinking. Usually as a scientist, you always think there's something you could optimize. So making a cut at some point and fixing the development and building many such instruments is really a bit of a challenge. But I think we did a really nice job, and we're still very happy with what we designed, a few years ago. So essentially, the Flamingo has this modularity. So you can recognize the two lenses that I talked about earlier. You have an illumination lens, a detection lens, a full illumination arm and detection arm, and you can have a total of two of both. So you can illuminate from two sides and you can detect from two sides. And so these modules can be added to the instrument and removed, and that also determines like how expensive the system is and what capabilities the system has. So it becomes very easy to build different configurations like a 2-lens, 3-lens, 4-lens, or LTX. But importantly, you can also take the entire system and turn it in space. You can turn it on the side, you can turn it upside down. And that offers a lot of opportunities in further tailoring the system to the sample and to the experiment. So just to give you an example, in the standard setup, we normally have a hanging sample, and these samples are typically embedded in an agarose gel or are freely suspended in an FEP tube, which is a material that has the same refractive index as water. And having these samples hanging into the chamber also facilitates the multi-view imaging, because as they are hanging, you can actually rotate them nicely without deforming them and do the multi-view imaging. Now, one limitation is that inside these tubes, there's only so many samples you can embed. Most of the time, actually, there's just one sample, and embedding multiple samples can become a bit of a challenge. And so if you rather want to image many samples, then what you have to do in this case, you take this configuration, you remove the red parts, the parts highlighted here in red, and you take the entire system, turn it by 90 degrees, and add the green parts. And suddenly you have an instrument that is entirely different, but 99% identical. And so you're using exactly the same components. You just swapped out a few parts, and suddenly you're able to image not just one fish, but like 20 fish in a dish. And you are able to image things that are sitting in a Petri dish, and you don't have to put them into a tube. And that can also make the difference sometimes. So this is just to show that it becomes very easy to also change these configurations. So briefly, there's the usual laser lines, filters. We can have 1 or 2 camera stages, etc. We also do the multi-directional illumination. So there's a way for us to eliminate striping. So, which is something we've always done, and it's also a feature in the SPIM in the Flamingo. So striping can be an issue in light sheet, and we have found ways to very quickly pivot the light sheet. So if you start pivoting the light sheet, these stripes move around, and if you do it fast enough, it eliminates the stripes. And so that's also a feature of the Flamingo, and which is extremely valuable to reduce striping. So essentially, we've addressed this problem that we need to be able to build many microscopes. So we have a common framework, we're reusing the same parts, there's a lot of 3D printed parts, and most importantly, it's the same electronics, the same software. And we can also remotely control these instruments, but I come to that at the end. Now, how do we address these, all the collaborators that may now want to use the system? Well, interestingly, as it happened, the system is portable. And so it really allows us to share the instrument with more people. So we don't just have this, big fancy setup that sits there on an optical table that only we can use and nobody else can use. And that's really been the main dilemma of, well, custom-built instruments, but also a lot of commercial setups. And so as commercial setups are usually not tailored for specific applications, we can also suddenly collaborate with people that are generally left alone, that just don't have a commercial system that would fit their needs, right? And as I said, oftentimes you can read about cool instruments, but it's not easy to get access to those. And so that's why most people would be faced with a challenge of building their own microscope. And that's not something that, generally speaking, biologists can do, because they may not have a workshop, they may not have a 3D printer, and they don't have like laser safety in their lab, etc. And so the other thing that we have recognized is that biological samples don't travel well. And biologists like to do experiments where their samples are, like right on their bench next to the incubator. And so that's really what the Flamingo happens to address. It happens to be a portable microscope. This wasn't really a design criterion, but it just happened to be super small, very robust, easy to set up, and something that we can move around in the lab. And so essentially, this cartoon summarizes the whole journey we've gone through. So we've taken a system off the breadboard, off an optical table, turned it into something more universal, more flexible, modular for us in the lab. But we can even put this in a suitcase and travel with it and share it with collaborators. And that's something we've done over the past few years, and we can show that this really actually works quite nicely. So we've taken this to courses like at the Marine Biology Lab in Woods Hole. We've had this set up there for several courses now over the past few years. We've taken this to UCSF, UCSD in California. The picture here on the top right is at Harvard. So we've actually shown that the system is portable and robust to be operational in remote places outside our lab. So this just to indicate that there's not just the microscope, but there's also a box that contains all the electronics, which is again a challenge on its own to combine all the electronics, power supplies, controllers, lasers into one portable box, so that you don't just have, lots of cables around the instrument, but everything is nice and compact, and also making it easier for the person at the receiving end to put it all together and have this operational. This also is very much in line with a perspective that we published a few years ago, which is talking about this challenge of putting advanced microscopy in the hands of biologists. And the summary of this article is basically depicted here, which is this idea that the engineers on one side built a cool microscope that they can then share with the biologists and iteratively move it back and forth to optimize the instrument. So as I said earlier, the engineers are not just sitting in the basement of their lab, like building cool instruments, but they're actually exchanging these with the biologists and getting their feedback to further optimize these systems. And that's just so valuable in trying to build useful instruments. Right? So we've imaged a lot of different samples, and this really suddenly allows us to also get out of our comfort zone and not just image some zebrafish, but really go out there and image some organisms that maybe haven't really been imaged successfully, because people haven't really had the chance to get their hands on a high-end system in those locations where those samples exist, right? And so suddenly we find ourselves traveling with these microscopes to places that are just not typically don't have access to high-end microscopy. So a lot of the customization really happens around the sample chamber, which is typically 3D printed. So we have different chambers, and together with the biologists, we really try to optimize how the sample is held in the microscope. And that is ultimately what really makes the difference and what decides whether the experiment is successful or not. We also now started having non-model organisms in the lab. So Platinareus is one upcoming model organism that we image in the lab. And that's just been exciting to also understand what exactly do you need on top of the microscope to actually be able to image something that you've just collected from the sea or picked up and is not fluorescent by itself. There's maybe no transgenics. So you also need to cover all the rest ahead of the actual imaging experiment, namely finding ways to anesthetize the sample, to mount it, to stain it, and to then image it. And so that's really another exciting area before you can actually start with the imaging. So a lot of Flamingo instruments have been customized and built, and here are just a few examples highlighting some of the parts that were built onto the system to allow unique experiments. So in the last few minutes, I just want to quickly also emphasize that you can also build a Flamingo for clear tissue imaging. And while this may not look like a Flamingo, it's more like a breadboard system. By still using the same components, we can still reuse the same parts and the same software, most importantly. And so that really allows us to build new configurations very quickly. And so this is a system that was specifically designed for imaging clear tissues at isotropic resolution. And just very briefly, in light sheet microscopy, the ultimate resolution is determined by your detection PSF, which is a wide field PSF, your light sheet profile. If you multiply the two, you can improve the axial resolution quite well. And this has mainly a prominent effect in the low NA case, where you can really severely enhance the axial resolution. But in order to really get isotropic resolution, you need to have the same NA on the illumination and on the detection side. So if you do so in the high resolution case, so NA of 0.3 is high NA in light sheet, you end up having a very short light sheet. And this would never allow you to image a large field of view, but you would end up with isotropic resolution. And so in order to make this useful, what you have to do is you have to scan this light sheet back and forth and synchronize it with the rolling shutter of your camera. So you're only seeing the thin part of the sheet as it's being scanned across the field of view. And then you can get the perfect light sheet, a thin light sheet that maintains the same thickness across the entire field of view. And there's a few other tricks that we do. So for example, we have this meniscus lens here on the illumination side. We have this remote focusing unit here. This is all explained in this recent preprint. And this system, this is depicting the meniscus lens and also a concave mirror we use to correct for field curvature. And also a real-time feedback system on the remote focusing unit. But ultimately, what this gives us is isotropic resolution. And that's really the holy grail in microscopy. And it's just phenomenal to see the quality of the data where you can really depict a centimeter-sized specimen, and you can rotate it in space, look at it from different directions, and you can't tell from which angle this specimen was recorded. So in the end, I just want to show you that the microscope itself doesn't come with any computer. So you have to use a laptop to remotely connect to a Flamingo and run an experiment. And that, of course, also means that you can run multiple experiments remotely from afar and run experiments where you gather data from different instruments, from different samples at maybe different resolutions, and then create a feedback loop where ideally in real time you're analyzing the data and making changes to your experimental workflow. And again, this is sort of along the lines of smart microscopy, but it has many more further implications. And some of that is illustrated here. So in the simplest case, we take the Flamingo and go to other places to be able to teach people about light sheet microscopy, right? We can also take the instrument to a remote place and do field work. We can image specimens on the beach, so to speak, right? You can set up a Flamingo in your Airbnb and be in a remote place, collect samples, and then image them. And the remote control aspect helps you in helping people do the experiment, right? The microscope needs to be where the sample is, but the person mounting the sample doesn't necessarily need to be a microscopy expert. It's somebody remotely can do the experiment. And that's also the idea behind this concept, where maybe in one place you have many flamingos and you have many samples, and people don't actually have to do the experiment in their lab, but they can do it remotely, right? So let's say you're a zebrafish scientist and we happen to have the transgenic line you want to image. We could, in principle, mount the specimen for you in one of our flamingos, and you can remotely log into my lab and run an experiment. Maybe while we are not using the instruments, maybe at night in our time zone, you can log into the machine and run an experiment. So you don't even need to buy the microscope or have the sample. As long as the microscope is where the sample is, you could remotely run an experiment. And another model could be that one person with one Flamingo travels the world and images specimens wherever they can be found, whether it is like in nature or whether there's a lab that happens to be the world expert in a certain species. You just go there with your instrument and image the specimen, and then, you know, ideally like stream all the data into a cloud and people collaboratively analyze the data. So I'm not saying we're doing this now. I'm just saying the Flamingo is prepared for all this. Okay, so this is what it looks like when we build microscopes, when we teach people about these instruments, and when we share them with other scientists. So with this, I'd like to thank people in the lab and acknowledge funding sources and ask you for your questions, and also emphasize that we are still recruiting as always, and looking for creative minds. So if you're interested in this kind of work, please reach out to me. And of course, if you're interested in having a Flamingo one day, we can also talk about this. Reaching other communities in other countries, sometimes it's easier to maybe have a lab that we could collaborate with that would be maybe able to build flamingos, so we don't have to ship them all the way from Europe to Australia. So there's a lot of challenges and opportunities that we have to think about in the future, how to disseminate more flamingos if that's what we want to do in the future. So thank you so much for your attention.
Thank you, Jan, for this incredible talk. That seems like a dream. One microscope to rule them all. That's yeah, no, that's fantastic. There is no questions yet, so I will come up with my own. What's the size limit? What's like the biggest thing that you have put in your microscope?
Yeah, I think I mean on the clear tissue front, samples can be centimeter size. So Here you can see a mouse brain, which is, I don't know, like a centimeter or so in diameter. So it, in the end, all depends on your working distance, and of course, a little bit also the resolution that you ultimately want to achieve. In this case, as you can see, this is a chamber with glass windows, and the detection lens is a dipping lens, but the illumination lens is an air lens. So if your sample ends up being really big, you can also move the lenses out of the way and use lenses with a long working distance. You just have to understand at that point, maybe the optics are less optimal. But as I said at the very beginning, the whole thing is pretty scalable. I think these water dipping lenses that we primarily use, like shown here on the left, these have a working distance of 3 millimeters, which generally for the live imaging is plenty, because as samples get bigger than that, they would typically also be just too opaque to be imaged. So most of the time, samples are a millimeter or a little bit beyond.
Thanks. And in terms of do you provide also support in terms of the image analysis? Because sometimes it's really nice you get lots of data, but then if people don't know what to do with them.
Yeah, of course we help people, with their data needs in terms of like storing and managing the data. It's not something where we necessarily have the perfect solution for everybody's needs, because they are all different. And in fact, it's yet another challenge that where there's probably other people who are, better suited to to solve these problems. Our expertise is more on the optics, on the microscopy. But yeah, obviously we don't leave people alone with these challenges. One thing to highlight maybe is that the instrument itself doesn't have any storage. And this is also to make sure that people think about their data storage needs first before they actually start the experiment, right? So on a lot of microscopes, you have this challenge that people come, they start an experiment, and light sheet is very, very dangerous, because you can image a specimen every 5 seconds from 4 directions for 24 hours, and then boom, you have 20 terabytes and you don't know what to do with it. And nobody can use the microscope until the data has been moved somewhere. So we force people to think about this first, purchase a data storage solution before they can actually run their first experiment. And so I think there has to be a conversation before the imaging, so that people are aware of their data storage needs and they think ahead a little bit about how they need to process the data. Because every Flamingo has the same data format, we would also hope that we can form a community around these Flamingos where people help each other with their data processing needs, right? So I'm sure there's a lot of people who can help out with processing the data, especially if it's sitting in a cloud and multiple people can have access to it, and find solutions how to process the data.
That's incredible. I mean, that's such a generous way of approaching microscopy and so unusual. It's it's quite mind-blowing. In terms of the illumination and the lasers, so how easy is it to set them up? Because I mean, there's always a lot of OHS set up around those things. So how easy is it for you to ship that and make sure that people are using them appropriately?
Yeah, it's actually relatively straightforward. And to our surprise, there haven't really been any major issues. So essentially, when you get a Flamingo, you have to unpack the microscope from its box, put it on a on a sturdy table, like a bench in a wet lab or so would even be good enough. Doesn't need to be an optical table. And then you have this electronics unit, and that's where the lasers are in, and the laser is fiber coupled. So essentially, in order to get the beam into the microscope, all you have to do is take the fiber and plug it in, and that's really the most delicate part of this process. Otherwise, there's a bunch of cables you have to connect. And so, we have pretty clear instructions on how to do this. And, sometimes one of us comes along and helps setting up the instrument. Sometimes we can, a manual is just good enough, or we can remotely assist people in setting up the instrument. So in terms of the laser, it's actually not the most difficult. Again, it's just that one fiber and then a few cables you have to connect. And that's never really been an issue. Also, in terms of laser safety, that's all enclosed. So, of course, it's a home built system, so no guarantee. It's not like a commercial system. But yeah, this really has never been a problem.
And how long does it typically take from the moment you receive the box to the moment you can start imaging?
I mean, we've done it in as little as half an hour.
Oh, wow. That's insane.
Sometimes it takes an hour or so, right? Because you need to, maybe move things around a little bit and prepare the table and find an outlet and hook it up to the network or set up the Wi-Fi. But generally, there's also no major realignment one has to do. So I'd say in an hour, you typically have the system up and running.
So it's really plug and play.
Yeah, I would say so. Yeah, and as I said, there's no storage and there's no real computer to run the experiment. So people would have to come with at least like an SSD. So on workshops, we typically bring like a 2 terabyte portable SSD, and that's good enough for most experiments. And then we all have like a MacBook that runs an app, the Flamingo app, and then we connect to the Wi-Fi on this system. It has a hotspot, and then we can run an experiment on the system for demo purposes or also for experiments. And and then sometimes we leave these instruments with collaborators for 2 or 3 months. And for some people, that's sufficient to do the experiment they always wanted to do. And we find that not everybody necessarily needs to buy a microscope and have it permanently. Sometimes people just need it for a month or two to do that one experiment, and maybe that's all they need. Right?
Thanks. And there's a quick question. What is the cost of building the microscope, roughly?
Yeah, so depending on the cameras and lasers, etc., it can be anywhere between 50 and 120,000 euros or dollars. So yeah, of course, it isn't as fancy as some commercial systems, and of course, that's just the cost of the parts. So compared to commercial systems, obviously that's a lot cheaper.
That is a lot cheaper.
And the cool thing also, because of the modularity, you could, for example, have multiple flamingos, let's say 3 different flamingos in 3 different configurations, but just one electronics box, right? And then and you could move the electronics box from one flamingo to another. Of course, you can only use one at a time, but it's the electronics like the laser, the stages, and the camera those are the most expensive parts. And if you can share them between instruments, that of course is a much more sustainable and more economic. Rather than having a laser on every single microscope, maybe especially if you're only using one at a time, that would be a waste, right? So this flexibility can also reduce the costs dramatically.
Well, thank you very much. Just before we finish, why Flamingo?
Yes, of course, I get this question all the time. It's not an acronym for fluorescence light something. I mean, the truth is it's just a cool name. So
It is, and it's very unusual. So that's I like the logo.
yeah, it's an unusual name, and hopefully a name that will stick, and people will remember how this cool microscope is called. It does look a little bit like a flamingo if if you look at it with its single leg, but
Yeah.
the name was there before the system actually took this shape. So yeah, simple answer is I think it's just a cool name.
It is a very cool name and a very cool system. Thank you so much for your time, and
Thanks a lot.
hopefully we can see more of your research. Thank you,
Yeah, thank you
and
so much.
thanks everyone. Bye.
Thank you. Bye-bye.